Welcome back to my course Aspects of Biochemical Engineering. In the last lecture, I tried to discuss the kinetics of substrate utilization, product formation and biomass production of microbial cells. And the process that we considered, that is the batch process, and also we discussed the Monod equation, and Monod equation is considered as a unstructured and unsegregated model, and I try to explain what do you mean by structured model, what do you mean by segregated models, and obviously that unstructured and unsegregated model is considered as a ideal model. It is not a real model. Real model will be structured and segregated model. And then we discuss about that how you monitor the concentration of the cells, and I told you two type of cells we have. One is called unicellular cell, another is what we call filamentous cells or multicellular cells. In case of unicellular cell, number is proportional to mass. So concentration of the cells can be expressed both either by mass per unit volume or number of cell per unit volume in case of unicellular cell. Now, in case of fungi or multicellular cell or filamentous cells, always we shall have to express the concentration of cell, which is used for explaining the enzymatic reaction kinetics. And we discussed what are the limitations of this equation. That I told you that mu is finite when S is finite, mu tends to mu max when S tends to infinity, but it does not explain what will happen to mu when S tends to 0. And in addition to that, there are two other limitations of this Michaelis-Menten equation, that it does not discuss the death of the cells, because we know any kind of living population always there will be growing of the cells and death of the cells. So, death of the cells is not considered in case of Monod equation, and in addition to that there is substrate inhibition and product inhibition as per microbial system is concerned, that also we do not consider. And then we discuss about the life cycle of the cells. I told you that life cycle plays very important role, because the reason is that in process we come across into the lag phase, log phase, stationary phase and the death phase. Every phase has a significance. As for example, lag phase we consider as the acclimatization phase, log phase is the active phase, and stationary phase is the starvation phase. And in case of death phase, that is the organisms are dying phase. So that. So why it is important? The reason is that whenever we prepare any kind of inoculum, or culture for any kind of fermentation process, we shall have to inoculate in between the mid log phase to late log phase. That is the culture we should prepare. Now, in addition to that, we discuss other different type of growth models that we recommended by different type of scientists, and also we discuss about the substrate inhibition and product inhibition, and also lutein pyrite model and PERT model. Now, today I am going to discuss that how this in the CSTR system, or when CSTR means continuous start tank reactor. When continuous start tank reactor we use in the biological system, we call it chemostat process. So, how in the chemostat process we can use the living cells. So, today's lecture will be mostly concentrated on that. Now, if you look at that this is a continuous process, we have I already told you that we have two type of process. We have one is your CSTR, what we call chemostat, another is the plug flow reactor. Now, chemostat basically this is simple a reactor, this is like this, and so there is a continuous inflow and outflow, and plug flow reactor is like this, that there is a tubular reactor and liquid coming one end and product is going out. Both are continuous reactor. Now, here we have uniformity in the reaction mixture, that by using the mechanical starter, here there is no starter. Not only that, during the movement there should not be any axial mixing. Radial mixing is permissible, not axial mixing. Now, fresh media is continuously introduced into the at a constant rate. So, we take it to fresh media here, and culture volume is kept constant in the by continuous removal of the culture. Now, suppose F is the flow rate. Now, and here also we shall have to maintain the flow rate F. Now, F, suppose this is F and this is F2, and if F1 is more than F2, then what will happen, that your volume of the reactor that keep on increasing, that is undesirable. And if F1 is less than F2, then volume of the liquid that will keep on decreasing with respect to time. So, this is the problem. Now, when F1 equal to F2, then and only then the volume of the reactor will remain constant. Am I right? So, this is the significance of this particular thing. Then we have a single nutrient control growth. We can what you call, now I told you the media comprises of n number of components. We have carbon source, we have nitrogen source, we have minerals, and we have vitamins. Am I right? So, you know that everything has some contribution to the living system. Carbon has 3 contribution. It goes for the cell mass formation, it goes for the source of energy, and it goes for the product formation. In nitrogen, mostly it is used for the growth of the cell. Minerals and vitamin, they mostly contribute as a cofactor in the metabolic pathway, the enzymes involved in the metabolic pathway. Now, so if you want to study that what is the effect of this component individually, we can easily study with the help of continuous systems. Just we can change the concentration here. Here we change the concentration, immediately we can find the changes here. The dilution rate, that is the rate of addition of phase media, determined by the specific growth rate, that is D equal to F by V. F is the flow rate and V is the, because the D is equal to mu, I shall show you in the next slide, dilution rate, because here I want to point out one thing, that when we study our life cycle, this is the X, this is the cell mass concentration, and this is time. So, we have this kind of that life cycle we have. Now, suppose in case of Baker's fermentation process, we are interested to operate it in the log. This is the log phase. Am I right? Because log phase we will get the maximum rate of cell mass formation. So, so that we can maintain by controlling the dilution rate by D equal to mu. That next slide, I hope you will get that information. Now, the continuous growth cell growth using the chemostat, as substrate continuously added and feed continuously removed, a quasi steady state is developed. Now, what I want to mean here, that in the chemostat process, that quasi steady state conditions means it is not exactly steady state, it tends to steady state. Now, suppose I told you, you can remember, whenever we operate any kind of continuous process, first we operate in a batch mode, and when your rate of cell mass growth is maximum, then we start feeding the media continuously and take out the fermented media continuously. So, this is like this. F is the flow rate, S0 is the initial substrate concentration, X0 equal to 0 when you have sterile feed. Sterile feed means there is no cell present in the media, and P0 means no product present in the media. So, here you will get, this is the F flow rate, S is the substrate concentration, X and P. Now, this we can have under steady state condition, and steady state condition, CO2 steady state condition is possible when and only when we operate the system for infinite period of time. Now, how we can do the cell balance? Now, we have the equation that rate of input plus cell generation equal to rate of output, accumulation of the cell and cell death. Now, here under steady state condition, that f into x0 is the cell that is input in the system. Cell generation is the, this is equal to what I can write dx by dt into v, am I right? Now, dx by dt equal to mu into x. So, I can write this is mu into vx. So, this is exactly what we have written here, that mu x into v, and this is the output f into x, because under steady state condition x is the series to substrate concentration, and this is the rate of cell that is the accumulation of the cell. Now, this will be equal to 0 and in under steady state condition, and this is the rate of death of the cells. So, quasi steady state conditions, the rate of accumulation that should be equal to 0, assuming the death of the cells is negligible and x 0 equal to 0. Now, this above equation I can write that this is, so this is equal to 0, and I assume this is also equal to 0. So, what will happen, mu x b equal to f into x. So, x will cancel, am I right? So, we can write this is mu equal to v by f. Mu equal to v by f means mu equal to d. So, what I was telling in the last previously, that by controlling dilution rate, it is possible to maintain a particular phase of growth for infinite period of time. So, this is very that is the advantage of this chemostat process. Now, compared to the batch process, we can refer that in the batch process this was the major drawback. Why? In the batch process you cannot hold a particular phase of growth for infinite period of time, but in the continuous system you can operate a particular phase of growth for infinite period of time. So, you can get maximum amount of cell mass formation. Now, if you do the substrate balance, previously it was cell mass balance. Now, if you do the substrate balance, then again we can write rate of substrate input plus rate of substrate generation, rate of substrate output, consumption and accumulation. So, we can write f into x 0 is substrate generation, means substrate cannot be generated in the reactor. So, this we can assume to be 0. This is the substrate output, this is the rate of formation, this is the degradation of the substrate, this will be plus, and under steady state condition this will be equal to 0. So, we can write f into s 0 into f s s equal to v into ds. Now, ds by dt, what we can write, ds by dt we can write ds by dx into, this is equal to dx by dt, am I right? We can write like this. Now, dx by ds we can write, this is 1 by x by s into dx by dt. This is what is written here, and then this is what has come. This is 1 by, and dx by dt equal to what? This is mu into xss. What is xss? Stands for steady state cell mass concentration. XSS stands for the, XSS is the steady state cell mass concentration. So, this equation we can write in this form, d into S0 minus SS equal to 1 by y x SS mu into x0. So, this is how we can develop the correlation between the steady state substrate concentration and the cell mass, steady state cell mass concentration. Now, next is a very important thing is that how this dilution rate affects the steady state conditions. So, this let me explain that. Now, let us assume this is we have, this is the start tank reactor, am I right? This is the start tank reactor, and so substrate is coming this way and product is going this way. So, here F is the flow rate, and this liquid volume we can consider as V. V is called the liquid volume, what we call working volume, because we assume that reaction take place only in the liquid phase, it does not take place in the free space. So, this is called working volume. So, this is also F. So, what we can do, I told you F by D, F by V, what is the F by V, what is the unit? This is volume per unit time, am I right? And this is what? This is volume. So, this volume will cancel and this will be time inverse. This is equal to dilution rate. This is equal to dilution rate. So, this dilution rate you can control, you can change the dilution rate as you, because V is constant. So, if you want to increase the dilution rate, you just increase the flow rate, volumetric flow rate of the liquid, then you can change that dilution rate. Now, if you see the correlation between D versus your XSS and SSS. What is XSS? I told you, this is the steady state cell mass concentration and SSS is the steady state substrate concentration. Now, this is steady state cell mass concentration and this is actually the steady state substrate concentration. This is SSS and this is XSS. So, you know that here you can see that at different dilution rate, this is D1, this might be 0, this is D2, this is D3, this is D4, this is D5, this is D6. But here what is happening? Here there is no cell present in the reactor, XSS equal to 0, am I right? XSS equal to 0. What you call, we consider this as a cell washout. What do you mean by washout of the cell? Because there is no cell present in the reactor, because your total reactor is free from the cells. Now, if there is no cell present in the reactor, what will happen? The S0, that S0 here and here also will be S0, because no cell, no reaction take place. So, this is exactly, this point will be S0. So, this corresponding point is this is called D washout. D washout. At D washout, there is no cell present in the reactor. Now, this is the major problem that we have in case of that chemostat process. Now, a question may be raised that why this happens. This happens due to the two different reasons. One, I can explain that we know the generation time. What is generation time? Generation time is generation time. Generation time is the time required for cell division. Cell division, time required for cell division. This is the time required, am I right? For cell division. So, suppose you are passing the cell in the, what is 1 by D? 1 by D is called HRT. What is HRT? HRT is called hydraulic retention time. Am I right? So, what is hydraulic retention time? That means how long you allow your liquid to reside in the reactor. How long that and why it is important? Because that is the time of reaction. Because the time you are allowing the liquid resides in the reactor, that is the time of reaction. Now, if your generation time and hydraulic retention time is less than the generation time, what will happen? Before cell multiplies, you are taking out the cell from the reactor. So, there is no cell present in the reactor. Another way it can be explained, that suppose this is a continuous reactor and you are passing one end like this. So, if suppose that here rate of growth of the cell dx by dt and rate of cell mass that is going out from this system, that is if rate of cell that is going out the system is more than rate of growth of the cell, then what will happen? The cell mass concentration in the reactor keep on decreasing and a time will come when there is no cell present in the reactor. Am I right? So, let us see that here what we have written, this is that we have already seen, this is mu equal to D. When mu equal to D under steady state conditions and the sterile feed. What is sterile feed? Sterile feed means here x 0 equal to 0. The input, there should not be any cell present in the feed. That is why we call it sterile feed. Then and only then mu equal to d. This is equal to, this is Monod equation, we know, mu max S K S. Now, previously we have done this the substrate balance, and in the substrate balance we have this equation. In this equation, if we put the value of mu, this is mu max S K S plus S, then we consider this as a chemostat model. This considered as a, this is called Monod chemostat model. The one Monod chemostat model is how it is done, because when we have this substrate balance or cell mass balance, and in the substrate balance and cell mass balance, and when we put the Monod equation, then we consider that a Monod chemostat model. Same thing we can work out in case of cell mass balance. Also we can find out that in cell mass balance also we can write the Monod chemostat model. Now let me show you how you can do that. Suppose this is a reactor and this is the input and there is the output. Now we have this reactor. This is F, this is x 0, this is F into x s s, and this is also x s s, and this is s s s. Am I right? This is V. So if you write the cell mass balance under steady state condition, what will happen? That the rate of accumulation equal to 0. Rate of input is F into x 0. Am I right? What is the rate of generation of the cells? That will be dx by dt into V. And what is the rate of output? F into x 0. X, not x 0, F into x. This is the rate of that, and then we can write this is and rate of accumulation, obviously that will be equal to 0. Now, if you divide this both the sides by V, this is V is there, we can divide by V, then what will have this be, the F by V equal to d into x 0, plus what is this, mu into x s s, what is this, V will cancel, and this will be d into x. Am I right? Now, this is equal to x into x 0 plus mu equal to mu max s s k s plus s s s into x s s. Am I right? Equal to, this is s s x s s, this is equal to d into x s s. Now, this equation also we call it Monod chemostat model. Monod chemostat model. So we have two Monod chemostat model. One is with respect to substrate balance, another is with respect to cell mass balance. Now, here the interesting thing is that the steady state substrate concentration and steady state cell mass concentration we can easily find out. Now, we have this equation, am I right? Now, if you look at this equation, that is mu equal to d equal to mu max s s s k s plus s s s. Am I right? So, this is equal to, I can write d k s plus d s s s equal to mu max into s s s. So, what I can write, s s s equal to d k s equal to mu max minus d. So, we can easily find out the steady state substrate concentration. Once we find out steady state substrate concentration, we can find out x value. How we can find out x value? Why we know y x by s equal to x s s minus x 0. This is equal to divided by s by s, s s s. Am I right? So, what we can write? This is x s s is equal to x 0 plus y x by s into s 0 minus s s s. So, if we put the value of s s s here, what we have done here, we can find out x s s value. This we can easily do that. So, this is exactly what we have done here. you see that this is x equal to this, so we can easily find out. Then if we multiply it by d, there is dilution rate, then what is d into x? This is d into x is equal to productivity, because already d into x equal to, we can write mu into x, and this is equal to dx by dt, and the dx by dt means gram of cell produced per unit time. This is called productivity. So, this we can write in this form. Now, when there is a very interesting thing, when you plot that d versus d into x, d into x means dx by dt, am I right? Rate of cell mass formation, and this what is the plot? Plot is like this. Now, in this plot here, what is this? This is equal to dx, this is maximum, am I right? So, what is dx maximum? Then at this point we call it dmax. So, what is dmax? Dmax means when the dilution rate at which the rate of cell mass growth, cell mass formation is maximum. So, this is what we can find out. Now, here in this plot, if you look at like this, so this is dx, am I right? This is into dx and then d. So, here I can write d dx by dd, this should be equal to 0. This is Plato, am I right? So, this is exactly if we look at here, here we have this dx equal to y x by s d s 0 this. Now, if we differentiate this with respect to d dd, if we do, then we will be coming across this equation. We will come across this equation, and why the dx, this is d is 0. This is the equation, and if you differentiate d, then it will be 1, this is a 0, and if we d square and this two term is there, so this will be equal to this value. And then we put this that d at when mu d stands for d equal tends to d max, dd by dx, that will be equal to 0. Then if you put these conditions, we will solve this and we find this equation. This equation we find, and finally we'll come across this equation, mu equal to mu max s k s y plus s. So this is what is this dmax? Dmax is the dilution rate when you will get the maximum amount of rate of cell mass production. Now, what is the maximum productivity? Now, if I ask you what is maximum productivity? Now, the productivity is usually expressed mass per unit volume. Maximum productivity means when the rate of cell mass formation is maximum. So, at Dmax your rate of growth of the cell is maximum. Now, at that particular situation, that whatever cell mass concentration is there, so but we have Dmax and XSS at that condition. If you multiply it by that corresponding steady state cell mass concentration, then we will get the maximum cell mass productivity. So, maximum cell productivity we can easily calculate. So, this is exactly, and from that we can find out the maximum cell mass concentration also x, since this is Dmax, that is so we can easily find out that maximum cell mass concentration, and this is how we can solve in this form. Final form will be coming in this. I told you this is d into x max, or this is we considered as a d max. If you have d max, we have x max, and if you multiply that, and this equation correspond to the maximum cell mass productivity. the maximum cell productivity can be calculated with the help of this equation. Now, that here we give you some graph, but what I have shown you that this is the dx, this is the x value, and this is the substrate concentration. Am I right? And this correspond to x 0. Am I right? So, so what is the S 0? Where at d washout, that S value will be S 0. Then what will be the d value at d washout value? D washout equal to d mu max into S 0 Ks plus S 0. So, we have seen that this is equal to d max. Am I right? This is equal to d max and this is the d washout. Now, when we want to produce more cell, we are interested to operate the system at d max. But you know that industry, how we operate the flow rate by controlling the valve. Now, if we increase the valve little bit more, the actually operator they do this work, but when we open little bit more, there is every possibility that d max can meet the d washout situation. So, this is the major problem that we have with the biochemical industry, that this problem can be overcome by two ways. Either you recycle the cell and or you immobilize cell on the solid matrix. This is the two. I shall come back. Now, if you look at this, that correlation between d max, d washout and mu max, usually the correlation is like this. D max, you can see this is d max will be less than d washout. Am I right? But d washout usually equal to or less than mu max value. This is the correlationship that we have. And if we want to now, we have shown you already how in the batch process we can find out the value of cell growth kinetics. Now, in case of chemostat also, it is possible to find out the cell growth kinetics if you write this equation in the form of line wave Bach plot. Then what we have to do? We have to plot 1 by d versus 1 by d, because everything is constant. What is this? This equal to y equal to c plus mx. Am I right? So, if you plot one is here, one with you will be having straight line. The intercept will give you the value 1 by mu max, and the slope will give you the ks by mu max. So, whatever mu max value you have, you put it here. So, we will get the value of ks. So, it is in the chemostat process also it is possible to find out the kinetic constant. Now, what are the advantages we have with the chemostat process? That log phase can be operated for infinite period of time. The effect of growth limiting substrate on the cell growth and morphology of the cell can be easily monitored, because I told you that cell comprises a number of components. Suppose we want to find out the effect of individual component on the particular process, that easily we can find it out. Several plant metabolites is usually produced during the transition of phases, which can be easily operated in the chemostat. Simple, by controlling dilution rate, we can have to and fro that movement of the phases. So, we can have more metabolite formation, and results obtained are reliable and reproducible. Major disadvantage of the chemostat is the cell washout problem, and growth over the long period can cause mutation and contamination. This is the major problem of this process. Now, as I told you that this drawback, that cell washout, that is the drawback, can be overcome by two ways. One is called chemostat with cell mass recycling and whole cell immobilization system. This I will discuss in my couple of lectures. Thank you very much.